Good morning, everyone, and welcome to today's presentation. I'm Dr. S.K. Singh, and I'm excited to share with you insights into the qualification of HPTLC, which stands for High Performance Thin Layer Chromatography. In this session, we'll explore the fundamentals of HPTLC, its significance, and how it is qualified for various applications. Let's get started and delve into this fascinating topic together. Let's begin by exploring what HPTLC, or high-performance thin-layer chromatography, actually is. It's an advanced version of the traditional TLC technique, designed to provide more precise and reliable results. This method is used in a variety of important applications. For example, it's crucial in herbal drug analysis, helping us identify and quantify active compounds in botanical medicines. It's also used for impurity profiling, which is essential for quality control and safety. Additionally, HPTLC plays a key role in identity testing, stability testing of pharmaceuticals, and fingerprint analysis, where we obtain a visual profile of complex mixtures. Now, moving on to the advantages that HPTLC offers over traditional methods. One significant benefit is the ability to analyze multiple samples simultaneously, which saves time and increases efficiency. It also uses less solvent, making it a more environmentally friendly and cost-effective option. And perhaps most interestingly, HPTLC allows for visual chromatographic fingerprinting, so we can directly see the unique chemical pattern of a sample, aiding in quick identification and quality assessment. Overall, these features make HPTLC a powerful tool in the analytical laboratory, especially when precision, efficiency, and environmental considerations are important. It really exemplifies how advanced techniques can enhance our ability to analyze complex samples reliably and efficiently. Let's take a closer look at an example configuration to understand how the components work together. The plate used here is silica gel 60F254, which is a common stationary phase in HPTLC. For the mobile phase, we're using a mixture of toluene and ethyl acetate in a 7:3 ratio, which helps in achieving proper separation of the analytes. Detection is carried out at UV 254 nanometers, allowing us to visualize compounds that absorb UV light. Now, moving on to the HPTLC instrument components, a typical system includes several key parts. First, there's the sample applicator, often an automatic sampler, which ensures precise and consistent application of our samples onto the plate. Then, we have the TLC plate itself, which acts as the stationary phase where separation occurs. The development chamber is where the plate is placed to develop the chromatogram through solvent migration. Once the separation is complete, the scanner or densitometer analyzes the plate, capturing the spots or bands. The UV cabinet provides the necessary UV light for visualization, especially for UV-active compounds. Finally, all the data collected is processed by specialized software, allowing us to interpret the results accurately. Together, these components form a cohesive system that allows us to perform high-performance thin-layer chromatography efficiently and reliably. Let's talk about the regulatory requirements for instrument qualification. In a GMP environment, it's essential that all analytical instruments are qualified using a life cycle approach. This ensures they perform consistently and reliably throughout their use. The qualification process is broken down into several key stages. First, there's design qualification, or DQ, where we verify that the instrument design meets our intended use and requirements. Then, we move on to installation qualification, or IQ, which confirms that the instrument has been installed correctly and is ready for operation. Next is operational qualification, or OQ, where we test the instrument to make sure it operates according to its specifications. Finally, performance qualification, PQ, verifies that the instrument performs reliably over time and under actual working conditions. The main goal here is to ensure that the instrument consistently works as expected and produces reliable analytical data. This is crucial for maintaining quality and compliance within our manufacturing processes. So, Each of these stages plays a vital role in safeguarding the accuracy and integrity of our analytical results. Now, let's talk about some key activities in our validation process. First, we need to clearly define the analytical needs. This step is crucial because understanding exactly what we require from our instruments guides the entire validation effort. Next, we verify that the vendor's specifications align with our needs, ensuring the equipment can meet our performance criteria. We also have to check software compliance, specifically with regulations like 21 CFR Part 11, which governs electronic records and signatures. Compliance here is essential to maintaining data integrity and regulatory adherence. Another important activity is confirming the availability of proper documentation. Well-maintained documentation supports traceability and provides evidence of compliance throughout the life cycle of the equipment. Finally, we come to design qualification, or DQ. This step confirms that the instrument we're selecting actually meets our user requirement specifications, or URS. It's about making sure that the design aligns with what we need before moving forward with installation and operation. These activities are all interconnected and form the foundation for a reliable, compliant, and effective validation process, ultimately supporting the overall quality and integrity of our operations. Now, let's talk about installation qualification, or IQ for short. This step is crucial because it verifies that the instrument has been installed correctly. Essentially, we want to ensure everything is in the right place and functioning as it should before moving forward. Key checks include confirming the instrument model and serial number, so we know we're working with the correct equipment. We also verify the power supply to make sure it's stable and meets requirements. Software installation is another vital point, checking that the software is properly installed and configured. Environmental conditions are also assessed, because factors like temperature and humidity can impact performance. We review calibration certificates to ensure the instrument is calibrated and ready for use. Additionally, we look at example documentation, installation checklists, vendor certificates, and operating manuals to confirm all documentation is complete and correct. The main takeaway here is that all installation parameters must meet predefined specifications. If everything checks out, we can confidently proceed, knowing the instrument has been installed properly and is ready for qualification and use. This step ensures reliability and accuracy moving forward in our validation process. Now, let's take a look at some typical tests we perform to ensure our instruments are functioning correctly. First, applicator precision is crucial because it ensures that the sample or reagent is being delivered consistently and accurately each time. Next, scanner wavelength accuracy is vital for correct detection and measurement, as even small deviations can impact results. Similarly, scanner photometric accuracy is essential because it confirms that the instrument's light measurements are precise, which directly affects data quality. Plate positioning accuracy is another key factor. It guarantees that samples are correctly aligned, preventing errors during analysis. Finally, there's operational qualification, or OQ. This step is all about verifying that the instrument operates according to the manufacturer's specifications. Think of OQ as a comprehensive check to make sure everything is working as it should before we use the instrument for critical testing. By conducting these tests, we maintain confidence in our equipment's performance and ensure the integrity of our results, which ties right back into our overall goal of delivering reliable and accurate data. Let's take a closer look at this example of applicator precision. In this case, we performed 6 replicate applications of a standard solution. The purpose here is to see how consistent the applicator is when used multiple times. By doing this, we can assess the precision of the application process. Remember, precision is all about reproducibility, making sure each application is as close as possible to the others. This step is crucial because if our applicator isn't precise, it can lead to variability in our results, which impacts the overall reliability of our analysis. So, by examining these 6 replicates, we get a clear picture of the applicator's performance and can identify any inconsistencies that might need correction. This example ties into the bigger picture of quality control in our analytical procedures, ensuring our measurements are both accurate and reproducible. Let's take a closer look at the scanner wavelength accuracy test. This step is crucial because it helps us ensure that our scanner is measuring wavelengths accurately. When we talk about wavelength accuracy, we're referring to how precisely the scanner detects the correct spectral position of a sample. If the scanner isn't accurate, our results could be off, which impacts the reliability of our data. During this test, we compare the scanner's readings against known reference standards. This allows us to identify any deviations or errors. Ensuring the scanner maintains high accuracy over time is essential for consistent and trustworthy measurements. Ultimately, this test ties into our overall goal of quality assurance. Reliable wavelength measurements mean our data is solid, supporting accurate analysis and decision-making. So, regular testing and calibration are key to maintaining the integrity of our spectral measurements. Now, let's talk about performance qualification, or PQ for short. PQ is all about verifying that our system performs reliably during routine analysis. It's a critical step to ensure consistent and accurate results over time. Typically, PQ tests include several key parameters. First, resolution, which measures how well our system can separate different compounds. Next is RF reproducibility, making sure that the RF values are consistent across tests. Then, there's peak area precision, which confirms that our measurements of peak areas are precise and reproducible. We also perform system suitability tests to verify that the system is ready for analysis before starting. These tests are usually performed periodically, like monthly or with each batch analysis, to ensure ongoing performance. Overall, PQ helps us maintain confidence in our analytical system, supporting the reliability and integrity of our data over time. Now, let's take a closer look at how we calculate the retention factor, or RF. This is a key concept in chromatography because it helps us identify and compare compounds. The RF value essentially tells us how far a particular compound moves relative to the solvent front during the separation process. To calculate it, we measure the distance traveled by the compound from the baseline and divide it by the distance traveled by the solvent front from the same baseline. It's a simple ratio, but it provides a lot of useful information. Keep in mind, consistent measurement techniques are important to ensure accurate and reproducible RF values. Understanding how to determine the RF helps us interpret our chromatograms and make meaningful comparisons between different samples. This ties back to our overall goal of analyzing and identifying compounds efficiently and accurately. Let's look at an example of how we perform a resolution calculation. This slide walks through the key steps involved. First, we identify the parameters we need, such as the wavelength of the light and the characteristics of the optical system. Then, we apply the relevant formulas to determine the resolution limit. The important thing to remember here is that these calculations help us understand the smallest detail our system can resolve. This example ties back to our overall discussion on optimizing optical performance, highlighting how precise calculations guide better system design. So, as we go through this example, keep in mind how these concepts are crucial for achieving the clarity and detail we aim for in our applications. Let's move on to preparing standard solutions, which is a crucial step in ensuring accuracy in our analysis. When we prepare these solutions, we're essentially creating a baseline or reference point for our measurements. This helps us verify that our equipment is functioning correctly and that our results are reliable. Next, we'll discuss the linearity check, also known as scanner qualification. This is about confirming that our scanner or detector responds proportionally across a range of concentrations. In other words, if we double the concentration, the response should also double. Performing this check is important because it ensures our instrument provides consistent and proportional readings, which is fundamental for valid quantitative results. By regularly conducting these checks and preparing standard solutions accurately, we maintain the integrity of our data and ensure our entire process remains reliable. This step ties directly into our overall goal of quality assurance in our analytical procedures. Let's talk about documentation and data integrity, which are fundamental to ensuring reliable and trustworthy data in our processes. First, we follow the ALCOA Plus principles. These are the key standards for good documentation. Being attributable means every data point should clearly link back to the person who recorded it. Legible ensures that the information is easy to read and unambiguous. Contemporaneous means recording data at the time the activity occurs, which helps maintain accuracy. Original refers to keeping the initial record intact, and accurate means the data must truthfully reflect what actually happened. Now, beyond these core principles, there are additional requirements to strengthen data integrity. An electronic audit trail is essential. It provides a secure record of all changes made to the data, so we can track modifications and ensure transparency. Controlled Standard Operating Procedures, or SOPs, guide us consistently and reliably in our documentation practices. Instrument calibration is also critical. Regularly checking our instruments ensures their measurements are precise. Lastly, periodic requalification helps verify that our systems and equipment remain in proper working condition over time. All these aspects connect back to our overall goal. Ensuring data integrity isn't just about compliance, but about maintaining the quality and reliability of our data, which ultimately impacts the safety and efficacy of our products. Let's take a closer look at the key points on this slide. First, HPTLC qualification is crucial because it ensures instrument reliability ***. That means our equipment performs consistently over time, giving us confidence in our results. Next, it ensures accurate analytical results ***, which is vital for making sound decisions based on our data. And of course, it helps us stay *** compliant with regulatory guidelines ***, which is essential in GMP environments. Now, moving to the qualification lifecycle. This is a structured process that begins with design qualification, or DQ, where we define what we need. Then we move to installation qualification, IQ, confirming the instrument is installed correctly. Followed by operational qualification, OQ, to verify it operates as intended, and finally performance qualification, PQ, which proves it performs consistently under real-world conditions. Finally, I want to emphasize that proper documentation and calculations * are absolutely essential throughout this entire process. They're what make our laboratory GMP compliant, ensuring traceability, accountability, and quality in everything we do. So, thorough documentation isn't just a formality, it's a core part of maintaining integrity and confidence in our analytical work. For further support, please don't hesitate to reach out to us. We're here to help and ensure you have all the resources you need. Feel free to contact us with any questions or for additional guidance. And with that, I want to sincerely thank you for your time and attention today. Your engagement is truly appreciated, and I hope you found this presentation valuable. Remember, we're always here to support you on your journey. Thank you very much.